Assalamu alaikum students, this is Irfan sir here. I hope you are all good. Last time we have completed a chapter on enzymology. Now we will start another chapter, that is cell division and cell cycle. It is very important. Frequently the questions are asked in pre-medical entrance examinations like NEET, AIMS. So this is very important. So let us start cell division and cell cycle. According to modern cell theory, according to modern cell theory, cells arise from pre-existing cells. Cells arise from pre-existing cells by the process known as cell division, by the process which is called as cell division. Now, during this cell division, all the events which are taking place during this cell division, all the events which are taking place during this cell division together constitute cell cycle. So cell cycle is a series of events which take place during cell division. Now a typical human cell, if we take human cell here, human cell, it divides once in approximately every 24 hours. If we take yeast divides once after every 90 minutes. After every 90 minutes, yeast cell, a eukaryotic single cell, it divides after every 90 minutes. Now the total time taken by the cell to complete cell cycle or cell division, we call it generation time. We call it simply generation time, or you call it GT. We will take here an example to understand it properly. For example, there is a cell, right, and its GT is given, that is 1 minute. GT of the cell is given, that is 1 minute. Now you are asked to find the time that the cell needs to form 32 cells. How much time the cell will require to form 32 cells if the GT given is 1 minute? Let's understand this. This is very simple. This is a cell, it divides and forms 2 cells. Now it divides again and forms 2 cells. Similarly, this will divide and form 2 cells. Similar process will go on. For example, here, listen carefully, and this will also divide. Now if you analyze this carefully, this cell will take 1 minute to form 2 cells. Now the 2 cells will take another 1 minute to form, how many cells? 4 cells. Now the 4 cells will take another 1 minute to form how many cells? 8 cells. So definitely another 1 minute will be taken to form 16 cells. Another 1 minute will be taken to form, yes, 32 cells. So if we count 1, 2, 3, 4 and 5. So the answer here is it will take 5 minutes to form 32 cells. If we little go further, 32 cells will take another 1 minute to form 64 cells. So another 1 minute here. So 64 cell formation will require 6 minutes. I hope it's clear. Now we divide this cell cycle into 2 phases. We divide this cell cycle into 2 phases. One is called I phase and another is called as M phase or division phase. Let's understand. So we have here cell cycle. Cell cycle, we divide it into 2 phases. One is called I phase, interphase, and another is called M phase, or you call it division phase also. It's a real division phase. Or you call it simply I phase. I stands for interphase. This interphase is also referred as energy phase also. You call it also energy phase or also preparatory phase. Preparatory phase. This interphase, if it's a preparatory phase, is energy phase because in this phase the cell prepares itself. The cell prepares itself for the upcoming M phase or for the upcoming what? Division phase. So it is a preparatory phase. And if you analyze this phase, cell is in a diverse metabolic activity. So cell is in a diverse metabolic activity. Cell is having a diverse metabolic activity in this phase. Cell is busy in synthesis of biomolecules, synthesis of proteins, different types of RNA. So cell is at its peak in the metabolism in the interphase. One more important point, this interphase, this phase occurs between 2 consecutive M phases. Between two consecutive M phases, you will find a phase that is called as interphase. So cell prepares itself for the upcoming division phase. Interphase, it takes 95% of the cell cycle, so it's the longest phase, and remaining 5% is occupied by M phase or division phase, because cell prepares everything here, and then it is time for the distribution. It is time for the distribution between the two cells, and that is happening in the M phase or division phase. I hope it's clear. Now further, this interphase, it was Howard and Paley. They divided interphase into three subphases. They divided interphase into, yes, three subphases. Three subphases, it is G1 phase, number one. Number two is S phase. And number three is G2 phase. Right? These are the three phases of the interphase. Now first we will understand G1 as G2. Let me complete this first, and then we will go with the M phase or division phase. So let's first discuss interphase, right? So here interphase first, interphase, and in this interphase we will first go through the G1 phase. G1 phase is simply, G stands for your growth, is a growth first phase, or you also call it pre-synthetic phase. S stands for synthesis, it is a pre-synthetic phase also. Now what's happening in the G1 phase? In the G1 phase, number one, there occurs protein synthesis. Number two, there occurs different types of RNA synthesis take place here, RNA synthesis. Metabolites are accumulated, metabolite pooling occurs, accumulation occurs. Like there is amino acids are pooled, nucleotides are pooled, they are pooled. And there is organelle duplication here, organelle duplication occurs, organelle biosynthesis takes place. There is membrane biosynthesis, very important, membrane biosynthesis occurs, right? Membrane biosynthesis occurs, organelle duplication occurs. And this phase, that is a G1 phase, it is the longest phase of the interphase. It is the longest phase. It's the longest out of the three, G1, S and G2. Out of the three, G1 is the longest phase. So now, in brief, we have seen here protein synthesis, RNA synthesis, metabolite pooling occurs, organelle duplication occurs, membrane biosynthesis, and this one is the longest phase, that is the G1 phase. So this is all about the G1 phase. Now there is S phase. S phase. Let us first see now the S phase here, right? S phase. S stands for synthesis, so it is a synthetic phase. So there is S phase now. S phase, you also call it post G1 phase because it occurs after G1. You can call it post G1 phase. You can also call it pre G2 phase because it occurs before the G2 phase. You can call it pre G2 phase. A very important point. First important thing you must understand, in this phase there is DNA replication taking place. DNA replication occurs in the S phase. This is the first important point to understand. DNA replication occurs in S phase. Number two, there is centriole duplication. Centriole duplication occurs in the cytoplasm in S phase. DNA replication in the nucleus and the centrioles get duplicated in the cytoplasm. Listen very carefully. Listen. Generally in an animal cell there is a pair of centrioles present. There's a pair of centrioles present in an animal cell. Now once they duplicate in the S phase, it will result into another pair of centrioles. So how many centrioles in total? 4 centrioles are there, or there are 2 pairs of centrioles. Now listen carefully, listen very carefully. Now this pair of centriole together with the surrounding cytoplasm, you see this is the pair of centriole and this is the cytoplasm. The pair of centriole together with the cytoplasm, this is called centrosome. We call it centrosome. Similarly here, there is a cytoplasm surrounding the centriole, and this is another, yes, this is another centrosome. So very clearly, so there is a pair of centrosomes, right? There is a pair of centrosomes, and there are two pairs of centrioles, or there are four centrioles. Generally there are two centrioles, but in the S phase there occurs duplication resulting into another pair that is equal to two pairs of centrioles, and two centrioles plus the surrounding cytoplasm is centrosome. This centrosome, they are important, very important for the spindle formation. They are very important for the spindle formation. Spindle formation is basically depends on the here centriole. Got it? Now there's a third important thing which is going to take place here, and that is histone protein synthesis occurs here. Histone protein synthesis also occurs in this phase. We know histones, they are very important for the DNA compaction. They are very important for the DNA condensation. Histones provide the basic level, basic degree of condensation in all the eukaryotes, be it a single cell eukaryote or complex organism like humans. Histones provide a basic level of the DNA compaction or the DNA condensation. So histone protein synthesis, they are synthesized, or their synthesis takes place in the S phase. Now S phase, what's happening in the S phase? Quick review: DNA replication, centriole duplication, and there is a histone protein synthesis taking place in the S phase. Now very important point, students often get confused. If the replication of DNA takes place here, what about the number of chromosomes? This is very important. Even if there's a DNA replication taking place in the S phase, chromosome number will remain the same. Very important point, chromosome number will remain the same. Each chromosome will be containing double set of genes. Each chromosome will be containing the double genetic material, but the chromosome number will remain the same even if the DNA replication has taken place. I hope it's clear. Let's move quickly to the another phase of the intraphase, that is G2 phase. So let us see the G2 phase here. So here it is G2 phase. So accordingly, G2 phase can be called as post. It can be called as post G2 phase. G2 phase can be called as post S phase. Post S phase because it occurs after S phase. So it's a G2 phase here. G2 phase. In the G2 phase, there is protein synthesis continues here. RNA synthesis continues here. RNA synthesis, protein synthesis continues here, and organelle duplication also continues here. Like mitochondria division, mitochondria number increase here, ER synthesis occurs here, Golgi synthesis occurs here. So organelle duplication continues here, and the membrane synthesis also continues here. Membrane synthesis also continues here. So this is about the G2 phase. Now after completing this interphase, G1, S and G2 phase, there is another phase of the cell cycle, that is M phase, or that is called as division phase. Let's move towards the another phase of the cell cycle, that is M phase or division phase. In the M phase or division phase, let me first tell you very clearly, the cell division in the eukaryotes, mainly it is of two types. One is called mitosis, another is called as meiosis. One is mitosis, another is meiosis. Interphase for mitosis and interphase for meiosis is same. Same means here, interphase consists of three phases, G1S, G2 here, and G1S, G2 here, that we have discussed. Now we have to discuss mitosis and meiosis separately. Actually, we have to discuss the M phase of mitosis and M phase of meiosis. First, we will see M phase of mitosis, because already we have seen the interphase of the mitosis, right? So M phase of the mitosis, and this M phase, M stands for mitotic phase here. If we are discussing mitosis, and here we can call it meiotic phase. This M phase, we divide it into two phases. The division phase or M phase, we can call it division phase also. It is divided into two phases. One is called karyokinesis, and another is called, you know it very well, cytokinesis. Karyokinesis means division of nucleus, simply you people know it very well, and cytokinesis means division of the cytoplasm. But precisely speaking, the karyokinesis means it means basically the distribution of the duplicated chromosomes into two nuclei, means the nuclear division. The two nuclei will be formed if the chromosomes get equally distributed in the two nuclei. So this is the distribution of the duplicated chromosomes into two nuclei, or in simple terms we call it nuclear division. This karyokinesis, it is divided into four phases. Divided into four phases. It is prophase, it is metaphase, it is anaphase, and it is telophase. Sometimes the sequence is asked about these phases, so you can remember PMAT, prophase, metaphase, anaphase, and telophase. These are the four phases of the karyokinesis, and later on we will see the cytokinesis also. First, let us discuss karyokinesis. I hope it's clear. So karyokinesis here. In the karyokinesis, first we have to see prophase. Let's understand the prophase here first. Prophase. So we are discussing the karyokinesis, and in the karyokinesis we are discussing the prophase. Prophase. In the prophase, there occurs chromatin condensation. Chromatin condensation occurs in the prophase. Chromatin condensation means here chromatin undergoes compaction. Chromatin undergoes condensation. In the interphase, there was genetic material was present in the form of chromatin. Chromatin is loose, relatively uncoiled, uncondensed, and chromatin in the interphase is well suited for the replication, well suited for the transcription, because the genes are relatively uncoiled. But interphase is gone. Now it is time for the chromatin to undergo the condensation, to undergo the compaction. And once the chromatin undergoes compaction and condensation, it becomes chromosome. Now you call it chromosome. You can call it as a chromosome. Condensed chromatin, you can call it chromosome. And once it will undergo condensation or compaction, definitely the chromosomes will become short and thick. Definitely, if anything gets condensed, it becomes short and thick. So chromosomes become short and thick because they undergo the condensation. Next point, each chromosome is here seen. Very important point. Each chromosome is here seen to have two sister chromatids. Each chromosome is here seen to have two sister chromatids. Let me clear this. This was a chromosome earlier in the interphase. Genetic material is double, right? But it is still a single-stranded chromosome. It is still a single-stranded chromosome, and here it is a chromatin here. Now in the prophase, you will see there are two chromatids here. Why there are two chromatids? Because this chromosome has a genetic material for two chromatids. It has double quota of the genes there, double amount of the genetic material. That's why it splits into two chromatids. You call it sister chromatids. Chromatids of the same chromosome, it is called sister chromatids. These are the chromatids of the same chromosome. So this is also chromosome, this is also chromosome. But this chromosome you will see in prophase. In prophase, chromosome consists of two sister chromatids which are held together at what? At centromere. You know it very well. We have discussed it already. Centromere. This is one more important thing. The next very important point that occurs in the prophase, centrosome, that we have discussed. Centrosome begins to move towards opposite poles. Centrosome begins to move towards opposite poles. Now what does it mean, centrosome begins to move towards opposite poles? Let's understand this thing. This is a very important point to understand. Let me tell you. What does it mean? This is a cell here. Listen carefully. This is cell. This is a nucleus. And a pair of centriole here. A pair of centriole is here, right? Now what happens exactly here? This centrosome is here and this is a centrosome here. They move away from each other by radiating out microtubules. Again here. So what's happening? The centrosomes are drifting away from each other by giving out what? By radiating out what? By radiating out microtubules. So these are the microtubules. Microtubules are proteins. They are made of a protein called tubulin. You call it tubulin. So they radiate out. Initially this is called sunburst. This you call it sunburst. You call it sunburst. Why sunburst? Because as if it is a star giving out radiations, but this is not a star here, but this is simply a centrosome here. On one side there is one centrosome, on another side there is a centrosome. It radiates out microtubules as if it radiates, it is radiating what? Radiations. But it's not radiations there, they are the microtubules. They are not the rays, they are the microtubules made of a protein called tubulin. That's the reason you call it sunburst. So here is one burst here. You can call it, this is one burst here, microtubule burst here, and microtubule burst here on the two ends of the at the two ends. This is called as ester. This microtubule burst at one end and this microtubule burst at another, we call them as esters. We call them as esters. So there are two esters, there are two microtubule bursts, right? So the resulting spindle is called amphiestral. The resulting spindle is called amphiestral. Why amphiestral? Amphi means two, estral means there are two esters present at two ends. So there are two esters present at two ends. That's the reason in animals the spindle is referred to as amphiestral. So there is a microtubule burst at one end, there is a microtubule burst at another end. Hence we call them as we call it as amphiestral. So this was the centrosomes moving away from each other, radiating out microtubules. So we can say here that spindle formation starts here. Next important point, which you can draw from this, that centrosomes move away from each other. So spindle formation starts here. It begins here. Spindle formation starts here. It begins here. One more important thing. Listen very carefully. This is a chromosome here, right? This is a centromere here, right? In this phase, there is a deposition of a very specific protein here. There's a deposition of a protein here. And this protein is called a kinetochore. This deposition of kinetochore protein, it occurs in the prophase. And where it is deposited? It is deposited at the centromere. Kinetochore deposition occurs at the centromere, and its deposition occurs in the prophase. Now, what is the significance of the prophase? This kinetochore. Kinetochore, basically, it is important for attaching the chromosome with the spindle, with the microtubules. Kinetochore helps the chromosome to get attached with the microtubules. To be very precise, this is the kinetochore which gets attached with the microtubule, so the chromosome gets attached with the microtubule. So it helps in the attachment. I hope it's clear, right? So this was about the kinetochore. In the late prophase, you will see nuclear envelope. This nuclear envelope, Golgi apparatus, right? And ER, endoplasmic reticulum, and nucleolus. They all disintegrate. They disappear. So they all disintegrate, they disappear. In the late prophase, nuclear envelope, Golgi apparatus, ER, and nucleolus, they disintegrate. So it means inside the chromosomes are in this fashion, in two-stranded form, right? And now what happens? In the late prophase, there is a disintegration of the nuclear envelope, Golgi apparatus, ER, and nucleolus. So this is about the prophase. So in the prophase, what happened? There is chromatin condensation. Next, chromosome becomes thick and short. Then chromosomes become thick and short, and next is centrosomes begin to move towards opposite poles. Spindle begins to form, and there is a deposition of kinetochore at the centromere. And at the last, what's going to happen? There is nuclear envelope, Golgi, ER, and nucleolus gets disintegrated, right? Now the disintegration of the nuclear envelope marks the beginning of another phase of the M phase, that is metaphase. So now there is metaphase. So second phase is metaphase. So in the metaphase, which marks the beginning of the metaphase, disintegration of the nuclear envelope marks the beginning of the metaphase. Now in the metaphase, spindle formation is complete. Spindle formation completes. And another important event, chromosomes get aligned, all the chromosomes get aligned at the center or equator of spindle. All the chromosomes get aligned at the center of the spindle or equator of the spindle. Let me explain this. This is important. So two important events here: spindle formation completes and chromosomes get aligned at the central plane or at the equator of the spindle. Listen carefully. See what happens exactly here. This is a cell here. Nuclear envelope is gone. This is a pair of centriole. This is a pair of centriole. Now at the center of the nuclear region, there are chromosomes scattered. This is very precise. Listen carefully. Very important. Now we know already that there is a radiation of microtubules from the centrosomes. Let me draw it with another color. So this is a pair of centriole, this is a pair of centriole. Radiate out, yes. Radiate out the microtubules. So they grow. The microtubules which are radiating out from the centrosome, they grow through the central region of the nucleus, where what is present, where the chromosomes are scattered. This is the important point to understand. The microtubules of the spindle grow through the central region of the nucleus, where the chromosomes are scattered. On their way, when they reach at the center, definitely they will have to face the chromosomes, because chromosomes are at the center. Now what happens really here? Listen carefully. So this is for example a microtubule is growing here, right? It's a microtubule growing. Now what happens here? It comes in contact with the what? With the kinetochore. I told you. It comes in contact with the kinetochore. Once it comes into contact with the kinetochore, what comes into contact with the kinetochore? The microtubule. Once it comes into contact with the with the kinetochore, this microtubule, it gets stabilized, it gets caught. I repeat, microtubule is stabilized and is caught. It is caught by the kinetochore. And same thing occurs on the other side. So what is happening here? So microtubule is growing from the other side also, right? So again here they are caught, right? So they are caught. Similarly, there is one more chromosome here, and this red is a kinetochore providing attachment to the chromosome, and the microtubules are caught here. So what happens really here? Similarly, all the chromosomes, they get eventually caught by what? They get eventually attached with the kinetochore, with the microtubule of the spindle. And the microtubule which are growing through the center of the nuclear region, on their way they come in contact with the kinetochore of the chromosome. They get stabilized, microtubule gets stabilized and get caught, and eventually chromosomes get attached with the spindle, and this way the chromosomes will come on the spindle at the center, right? And this is called, this central region, it is called a metaphasic plate. It is called a metaphasic plate or equatorial plate, right? It's called a metaphasic plate or the equatorial plate. Now they are brought at the center. Now why they are brought at the center? So that to have an equal pulling force on the two sister chromatids. In the next phase we will see that there is a pulling force acting on the two sister chromatids. If they are brought at the center, the force will be equal, right? So they will split equally. But if the length of the microtubule, or if they are not brought at the center, if the chromosomes are not brought at the center, the pulling force will be unequal. Definitely the two chromatids will go towards one pole only. They will not split. This is the reason why they are basically brought at the center. So this is about the metaphase here. But very important thing you need to understand here. In the metaphase, you see that chromosomes individually are seen on the spindle. Chromosomes are seen as discrete elements present on the spindle. That is the reason chromosome morphology is best studied in the metaphase. So if you want to study the chromosome morphology, remember that it is very much asked in your exams. It is best studied in the metaphase. What is the reason? Why in the metaphase? Because in the metaphase, the chromosomes are well distinct. Chromosomes can be spotted. You can spot them individually on the spindle. They are present as discrete elements on the spindle. That's the reason you can study chromosome morphology best in the metaphase. I hope it's clear. And now once they come on the spindle, there is another phase now going to take place, that is called as Anaphase. That is Anaphase here. That is Anaphase. In Anaphase, what's going to happen here? Listen carefully. I'll show you diagrammatically so that you will understand here. So this is a spindle here. Microtubules, astral microtubules here. So let me show you 2 chromosomes at least. So this is one and this is another. This is a metaphase diagram actually, but I will show it on this so that you will understand. This is a kind of a core, and they get attached with this, right? So this is a metaphase diagram here. Metaphase. We have discussed this. Now in anaphase, what's going to happen? In anaphase, all the chromatids split simultaneously. All the sister chromatids of the chromosomes, they split simultaneously by the equal pulling force from both the sides, from both the poles. So what's happening in the anaphase? Splitting. There occurs splitting of which chromatids? Sister chromatids. There occurs splitting of sister chromatids in all the chromosomes, and simultaneously, synchronously, the splitting takes place. Now once they split apart, let me show it like this. So this is a chromosome here. This is important to understand here. Let me show it again here. So this is here. So what's happening here? The sister chromatids are splitting apart, and they split apart in such a way that their centromere is at the leading edge. Please try to understand. The chromatids are moving towards the opposite poles. Let me tell you again. In anaphase, there occurs splitting of sister chromatids. Once they split apart, now these are called chromosomes. Now these chromatids, once they apart from each other, you call them as chromosomes, because each chromatid has got its own centromere. If you want to count the chromosome counting depends on the number of the centromeres. Now each chromatid has its centromere. Now this is called what? This is the chromosome. This is no more a chromatid here, this is a chromosome here. Now they move in such a way that their centromere is at the leading edge. You see, centromere is at the leading edge, and the arms of the chromosomes are trailing behind. They are left behind, they are trailing behind. This is many a times asked, how chromosomes basically move in the anaphase towards opposite poles. They move in such a way that their centromere is at the leading edge and their arms trailing behind. So they move in such a way. What's their centromere? Centromere is at the leading edge. And what about the arms of the chromosomes? Arms of the chromosomes are trailing behind. Arms of the chromosomes are trailing behind. They're left behind. You got it? This is how they basically move towards the opposite poles. And what happens towards the end of the anaphase? All the chromosomes, now they were the sister chromatids, now called chromosomes. They have reached at the end of the anaphase. All the chromosomes have reached at the opposite poles. Now here we can say that in anaphase there occurs chromosome duplication. Chromosomes get double here, right? The number of the chromosomes double here, because now earlier it was one chromosome. Once they separate apart and each having its own centromere, you call them as chromosomes. So the chromosome number gets doubled in which phase? In the anaphase. So towards the end of the anaphase, what's happening here? So this is no more metaphase here. This is now anaphase. Got it? So this is the anaphase. Now during this phase, there is one more important aspect to understand here: the shape of the chromosomes. Let's understand that aspect. Shape of the chromosomes. Let me tell you. This is here type of chromosome. I will write here type of chromosome, and I will write here shape. Right? Type of chromosome. First is there is a metacentric chromosome. Metacentric chromosome. Metacentric chromosome means the centromere is at the center. Centromere is at the. This classification is based on the position of the centromere. This classification is based on the position of the centromere, and in the metacentric chromosome, the centromere is at the center. There is metacentric. There is submetacentric chromosome, and here we can write away from center. So here the centromere is away from the center. The third type is called acrocentric, and here where is the centromere present? It is present just below the terminal end, right? And the fourth type is called telocentric chromosome. Telocentric chromosome, what's there? The centromere is at the end. It is at the terminal end, or it is at the end. So this is the classification based on the position of the centromere and the chromosomes. Let me show it very clearly. This is very simple. So you can see these shapes during the anaphase. Remember that. If you just pull anything at the center, definitely the shape will be V shape. So here is the center. So what is the shape? It will be the V shape. And submetacentric, so it will be J shape. So here it is submetacentric, away from the center. So it will be L shape, not J shape here. It will be L shape here. And now acrocentric means just below the terminal end. It will be here J shape. So J shape here, and at the terminal end it will be I shape. You can understand. So there are shapes observed in the chromosomes during the anaphase, during their movement towards the opposite poles. So metacentric chromosome means centromere is at the center, and once you pull this chromosome, what's the shape of the chromosome during the movement? It is a V shape. Submetacentric chromosome, where the centromere is away from the center, if you pull this kind of a chromosome, definitely it will be L shape here, the centromere L shape. Acrocentric means the centromere is just below the terminal end, definitely it will be J shape. And the telocentric means the centromere is at the end, and if you pull this centromere, this kind of a chromosome, the shape will be I shape. So these are the shapes which are found during the anaphase of the chromosomes. Now the last important phase, that is telophase. We can say that telophase, let me write here first, telophase. In the telophase, there occurs DNA decondensation. What has happened in the prophase, opposite to that, we can say takes place to a large extent in the telophase. So there occurs DNA decondensation. DNA decondensation means DNA again becomes loose. It again becomes extended. It again becomes what? Chromatin. So we can say that the cell will enter into what? Back into what interphase? Nuclear envelope, Golgi, ER, nucleolus, they are reformed. They are reformed back. Right? So the DNA decondensation occurs, chromatin formation again here, right? Cell enters into interphase, because in the interphase there was genetic material present loosely, relatively less coiled, decondensed, that is a chromatin. And the nuclear envelope, Golgi, this nucleolus and this nuclear envelope, ER, endoplasmic reticulum, they all get reformed back. Now, if you just try to understand one more important point, very important point to understand here. Listen, there's a nucleus here. There's one nucleus formed, and there's another nucleus formed. Nuclear envelope is back, and these are the chromosomes. Now, these chromosomes are single-stranded. These are the chromosomes. Now, in the anaphase, chromosomes tend to collect in mass at the ends. means chromosomes will come in mass at the poles. In the metaphase, they were you can spot them individually on the spindle. So it means in the telophase you can say that this discrete nature, the individual nature of the chromosome is lost here during the telophase, because they collect in mass at the ends. And one more thing, important thing is there, the DNA decondensation occurs, chromatin becomes thin, it becomes thread-like, and eventually, if you are just basically observing it under the microscope, it will disappear under the microscope. Why? Because it becomes too thin to be spotted by the microscope. You understand this thing? This is important to understand here. Well, number 1 point, two things are here. Chromatin formation takes place, DNA decondensation occurs, uncoiling occurs, this condensation is removed, it becomes loose, it becomes relatively less less coiled, and due to which it becomes again thin, it becomes fully extended, due to which it disappears from under the microscope. And this is the reason that the individuality and this discrete nature of the chromosome is lost. So we can write here discrete nature or individuality of the chromosome. Individuality of the chromosome is lost because they tend to be collected in mass at the poles, plus the chromatin formation takes place due to decondensation and uncoiling. So this is about the telophase, and this is all about the M phase. And which part of the M phase? That is karyokinesis. Now another part of the karyokinesis is, another part of the M phase is cytokinesis. This was about the karyokinesis. Now there is another important part of the M phase, that is cytokinesis. Cytokinesis. There is a difference in the cytokinesis in plants and in the cytokinesis of the animal cell. In animal cell, cytokinesis occurs due to plasma membrane indentation or groove formation. Indentation, or we can write groove formation. What does it mean? So this is a cell here. This is one nucleus, this is another nucleus. These are the chromosomes inside, right? So this is a chromosome here. Now what happens here really? The plasma membrane, it shows indentation, or it grooves. It goes inside like this, and there's a formation of a groove and this indentation. And this groove, it basically grows further. It furrows deep. There's a formation of a furrow or a groove. It grows further, it furrows deep. Eventually, the two ends meet and result into the formation of two cells. Right? So this furrow or this groove, it grows deep, and the two opposite ends of the plasma membrane, they fuse, meet, and eventually resulting into the formation of two cells. And this, may I tell you one important thing here. Listen carefully. This indentation, which is going to pull the plasma membrane, there's a point. Number 1 point, there are remnants of microtubules of spindle present here. Remnants left over. This is called a mid-body. We call it mid-body. Mid-body acts as a bridge between the two cells here. Number 1 point. Number 2nd point here is, there is a contractile cytoplasm. Contractile cytoplasm, which is present just below the plasma membrane, where the groove or the furrow formation has to take place. There is a contractile cytoplasm, and there is a mid-body. Mid-body is a leftover part of the microtubule of the spindle. This contractile cytoplasm, it contains actin, it consists of myosin, right? Even microtubule remnants are there. This contractile cytoplasm, it pulls the plasma membrane from both sides, because it's a contractile cytoplasm. Contractile means here, the actin and myosin, they are the contractile proteins. That is the reason we call this stuff here contractile cytoplasm, because these two are what? They are actually the contractile proteins. They undergo depolymerization and they pull the plasma membrane from both the sides, eventually resulting into the formation of two cells. I hope it's clear. Now there is cytokinesis in the plant cell. Cytokinesis in the plant cell occurs not by the furrow or grooving or the indentation. It occurs in a different fashion. In the plant cell, is a plant cell here, right? It's a nucleus here, it's a nucleus here. There is formation of the cell plate. There is a formation of the cell plate. Cell plate formation occurs in the plant cell, and it grows from the center towards end. It grows from the center towards end means there is a centrifugal fashion of the cell plate formation. There is a centrifugal fashion of the cell plate formation, and in the animal cell there is a centripetal because it grows towards from ends, from periphery towards ends. So in the animal cell it was, yes, what it was? It was centripetal towards center. And what is here? The cell plate grows from center towards end, so it is centrifugally. It is centripetally here. This is also asked, remember that. Now this cell plate formation, remember that this cell plate is regarded as, it is regarded as extracellular wall. This is important. We call it extracellular wall. Extracellular wall formation occurs here. Number one point. Number second point. Cell plate formation, it's the formation of the cell plate. It is first seen. The sign of the cell plate formation is first seen in the late prophase. In the late anaphase. In the late anaphase. In the late anaphase. The cell plate formation is first seen. Its first sign is seen in the late anaphase, in the formation of phragmoplast. Phragmoplast. Phragmoplast, it consists of microtubules. It consists of microtubules, number one. It also contains even actin elements are there, and it also contains Golgi derived vesicles. Listen very carefully. Cell plate formation occurs as extracellular wall, and the sign of the cell plate formation is first seen in the late anaphase in the form of phragmoplast. Phragmoplast is actin, it is microtubule, and there are Golgi derived vesicles. And these Golgi derived vesicles, they contain the material for the cell plate formation. And what is the material? It is a calcium and magnesium pactate. Calcium and magnesium pactate is the material for the cell plate formation. So these Golgi derived vesicles containing the calcium and magnesium pactate, they are basically assembled at the plane across which the division of the cytoplasm has to take place. Listen very carefully. These microtubules which are present here, and actin which is present in, and the Golgi derived vesicles containing calcium and pactate. Microtubules serve as tracks for the Golgi derived vesicles to unload the material. This is a very important point, and this is an asked question. There are two types of questions are asked. Number one, which synthesize the cell plate? Answer is very easy, that is Golgi apparatus. Now, which determines the plane in which the cell plate is to be formed? It is a microtubule, because microtubule serves as tracks for the Golgi derived vesicles to unload the calcium and magnesium pactate here. So eventually what happens, the cell plate formation takes place, and this eventually leads to the formation of two cells, and this cell plate, it becomes middle lamella. Of the two cells. As we know, middle lamella, its composition is mainly calcium and magnesium pactate, and it is derived from actually the cell plate, and middle lamella acting as a cementing material between the two cells. Now this is recognized as one cell, this is recognized as another cell, and then this cell plate which is earlier formed, now you call it middle lamella, and this middle lamella acting as a cementing material between the two cells. So this is cytokinesis which occurs in the plant cells. Now you got to know why it's called extracellular wall, because later on this cell plate it becomes the part of the extracellular wall outside the cell. First it is formed inside a cell, and later, once its formation is complete, once it becomes middle lamella, you call it extracellular wall. This is no more intracellular, this is extracellular. Now for this cell it is outside, and for this cell it is outside. Earlier it was inside, but now this is outside. So that is the reason it is called extracellular wall. I hope it's clear. So this was about the mitosis, the cell division. In the next lecture we will see the M phase of meiosis. Very important. Meiosis questions are also asked from the meiosis. So see you in the next chapter with the meiosis. Stay blessed, stay home, stay safe. Allah Hafiz.